Following our discussion on endogenous and environmental requirements for seed germination, we will now shift our focus to various chemical means of promoting germination in seeds. We will begin with the plant hormones abscisic acid and gibberellins. First, we need to understand what plant hormones are. Plant hormones are also known as phytohormones. The word phyto is a Greek word meaning of a plant or relating to plants. Plant hormones or phytohormones are signal molecules that are produced within the plant at extremely low concentrations. They serve as chemical messengers that are produced in different parts of the plant and have a physiological effect on a target tissue that may be distant from the site of production. So even if you have a phytohormone that is produced in the roots, it can affect the shoots, the flowers, the leaves, and all other parts of the plant. Each plant cell is capable of producing hormones. These hormones control all aspects of plant growth and development, from embryogenesis to reproduction, including biotic and abiotic stress responses. Because they control or regulate all aspects of growth and development, they are also called plant growth regulators. So if you read a paper that talks about plant hormones, phytohormones, and plant growth regulators, they are all referring to the same thing. There are several plant hormones that have been already identified, but for the sake of our discussion on seed germination, we will only focus on five major classes of phytohormones, which include abscisic acid, gibberellins, cytokinins, ethylene, and auxins. Let's begin with abscisic acid, or ABA. This is the chemical structure of ABA. ABA is a weak acid that was discovered in the 1960s in abscising or falling cotton leaves, hence the name abscisic acid. ABA is a plant hormone that positively promotes stomatal conductivity and plant responses to both biotic and abiotic stresses. When we say biotic stresses, these are stresses that are caused by living organisms such as pathogens and pests. Abiotic stresses, on the other hand, are those that are created by non-living factors such as cultural practices, soil properties, and weather, to name a few. Let's take a look at an actual example of an ABA regulated response to an abiotic stress. In this example, we have drought stress. Plants produce their own food via the process of photosynthesis. Photosynthesis requires gas exchange, and this occurs in the leaves, specifically in tiny pores or opening called the stomata. Stomata have two kidney bean-shaped structures on either side called the guard cells. When plants are well watered, the guard cells are filled with water and are therefore turgid. When the guard cells are turgid, the stomata are open, and this facilitates gas exchange as well as water release. When plants experience drought or shortage of water, it produces ABA in the drought-affected organs like the roots, leaves, and even seeds. And this ABA travels to the stomata to signal that water is scarce. When the stomata receive the signal, water leaves the guard cells, and the guard cells become flaccid, like what you see in this panel. When the guard cells are flaccid, the stomata are closed. When the stomata are closed, the plant is able to prevent further loss of water through these openings. This is just one function of ABA. How about during germination? ABA is a negative regulator of seed germination. Even at very low concentrations of 1 to 10 micromolar, it actively suppresses seed germination. ABA can also interact with other plant growth regulators such as gibberellins, ethylene, cytokinins, and auxins to prevent germination. Let's now look at how ABA affects seed germination in Arabidopsis seeds. From previous lessons, you already know that Arabidopsis endosperm is composed only of a single layer of cells. This single layer of cells continuously synthesize and release ABA, which is transported to the embryo by the ABA transporters. Two ABA transporters that move ABA from the endosperm to the embryo are ABCG25 and ABCG31. Aside from the endosperm, the embryo itself produces ABA, and this ABA is locally transported within the embryo through the function of the embryo transporters ABCG30 and ABCG40. This slide illustrates the effects of ABA on seed germination in Arabidopsis. The first panel shows Arabidopsis germination after 0 hours, while the second panel shows Arabidopsis germination after 48 hours. The seeds that are labeled WT here, 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 and here are normal seeds. The seeds that are labeled ATABCG, followed by numbers, are mutant Arabidopsis seeds with non-functional ABA transporters. As I explained from the previous slide, the ABA transporters move the ABA from the endosperm to the embryo or specific locations within the embryo. These mutants are able to produce ABA in the endosperm and the embryo, but they are not capable of transporting ABA to where it should go. So if we look at the first panel, we see zero germination. And by zero germination, I mean no radical or embryonic root protrusion. This could be because the seeds have only been in germinating media for less than an hour, and it's not enough time for the seeds to germinate. In the second panel, we can see germination of all the seeds, but notice that the mutants have either more seeds germinated compared to the non-mutants, such as in the case of ATABCG25 mutant, right here, or faster rate of germination as in the case of the 3 other mutants. So what does this tell us about the role of ABA in seed germination? This data tells us that it is not enough that the seeds are producing ABA. ABA needs to be transported to specific organs or organelles. ABA as a signaling molecule tells the plants not to germinate. If the signal just stays in the endosperm or specific parts of the embryo where it is produced and not transported, then the seed would think there is no ABA and that means we can germinate. Now below we have graphs showing the germination of mutants and the wild type with or without stratification. Arabidopsis is a temperate species and requires pre-treatment in cold and moist conditions before it can germinate. Without stratification, we can see that the mutants germinated faster than the normal stratified seeds. The red, green, purple, and blue lines here represents the germination of the mutant lines. With stratification, we don't see much difference in the germination profile of the seeds, and this could be because during stratification, germination inhibitors such as ABA are leached out of the cell, allowing the seeds to germinate. Following ABA, we have gibberellin or gibberellic acid, or simply GA. This is the chemical structure of GA. Gibberellins were first discovered in Japan. A fungus called Gibberella fujikuroi infected rice plants and caused them to grow too tall and fall over. The fungus produced a chemical that stimulated the growth of the rice plants. The chemical was later isolated and was named gibberellin after the fungus. It was later found out that plants naturally produce variations of these chemicals. GA plays an important role in plant growth and development. It strongly promotes transition from the vegetative to the reproductive stage. It is required for pollen function during fertilization, and it promotes cell elongation. Perhaps the most important role of GA is in promoting stem elongation between nodes on the stem or internodes. A node is a place on a stem where a leaf attaches. Gibberellins induces elongation of the stem between the nodes. It is easiest to see the absence of gibberellin in dwarf plants and rosette plants. This panel here shows 2 sorghum plants. The one on the left produces GA, while the one on the right is defective in GA synthesis. You can easily see that without GA, sorghum was not able to elongate. But how does GA affect germination? The presence of GA is a requirement for seed germination. During this process, GA synthesis or production increases. The germination of seeds is stimulated by gibberellins through the release of seed coat dormancy, weakening of the endosperm, and expansion of the embryo cells. This slide shows you how reserves are mobilized in monocots via GA signaling. During germination, GA is released from the embryo axis, as well as from the scutellum, into the allurone layer. As you already know, the allurone layer is capable of synthesizing enzymes that can degrade stored reserves in the seeds. GA induces the synthesis of these enzymes in the allurone layer, which are then transported to the endosperm. These enzymes include proteases, amylases, and glucosidases. Once released into the endosperm, the enzymes degrade starch into sugars, which are then absorbed by the scutellum and then transported back to the developing embryo axis. This slide illustrates how GA affects seed germination in Arabidopsis in relation to ABA content. GA and ABA have antagonistic relationship in the context of germination. ABA prevents germination, while GA promotes germination. In this panel, we have the normal seeds labeled as WT or wild type. We have ABI4, which is a mutant that cannot produce ABA, and OE1, which produces too much ABA. The seeds were germinated in 1/2 MS media, 1/2 MS media containing paclobutrazol. Paclobutrazol inhibits GA synthesis. And then we have seeds that were germinated in 1/2 MS media containing GA. These graphs on the right are germination data that corresponds with the data taken from the pictures in the left. Let's focus on the germination of these seeds after 1.8 days in 1/2 MS media. In this graph, we can see the germination of normal Arabidopsis seeds as indicated by the red line. The germination rate of ABI4 was a little higher than the normal seeds as indicated by the black line. ABI4 are not able to produce ABA, which is a germination inhibitor, so naturally without ABA, the seeds will have better germination. OE1 showed lower germination compared to the normal and ABI4 seeds as indicated by the blue line. OE1 produces or overproduces ABA, and we know that ABA prevents germination, so naturally, if a seed is producing more ABA, it will have lower germination. Now in the second graph, we have a medium supplemented with paclobutrazol. Paclobutrazol, as I said, inhibits GA synthesis. GA is necessary for germination. Without GA, we can expect poor germination of seeds. Now, if we look at this graph, we can see an overall decrease in the germination of all the seeds compared to those germinated in plain MS media. Lastly, we have seeds that were germinated in MS media supplemented with GA. In contrast to seeds treated with paclobutrazol, all the seeds supplemented with GA showed better overall germination compared to seeds that were germinated in plain MS media.